Good evening. As Dean of the School of Medicine, it's one of the joys to work with such dedicated faculty or outstanding educators, mentors, and researchers. I want to welcome you all to the first of what I hope will be an annual Dean's Faculty Awards ceremony. My wish would have been to be with you in person, but like all things going on currently, things are different, and tonight this event will be virtual. It's been a challenging year, and as we start 2021, my wish and prayer for all of you is that you have good health, we see stability in this country, and we see an end to this pandemic. These times remind us, however, how important it is to recognize the positive things in our lives and to recognize our faculty, especially, who have been so dedicated to meeting the missions and advancing the missions of New York Medical College. Tonight, we honor and celebrate the excellence and dedication of our faculty who do amazing things here at New York Medical College. I want to thank the committee of your peers who selected the honorees, but I would be remiss in not saying congratulations to all of our faculty, and I know the decision was very difficult by our committee to select the winners. While we'll be naming certain faculty chosen tonight, I wanted to thank everyone for their attention, for being here, and continuing to support New York Medical College. Let's all enjoy this evening together. Thank you.
Thank you, Dr. Nadler, for that warm welcome. I'm Catherine Spunknabel, and I'm Senior Associate Dean of Faculty Affairs for the New York Medical College School of Medicine. I will be your host for this inaugural Dean's Faculty Awards virtual ceremony. Although we're unable to meet in person, there's plenty of opportunity for live interaction during the pre-recorded event using the QA chat function. The virtual ceremony program will also be made available on the college's YouTube channel for future viewing. Before we get going, I'd like to thank several people who made this event possible. First, many thanks to our very dedicated and talented Dean's Office staff, the educational media team led by Michael Cotter, and college administration for funding the award and honorarium. Many thanks to the more than a dozen department chairs who nominated their faculty, and in some instances each other, for award consideration. And many thanks to the Dean's Faculty Awards Review Committee. The committee did a tremendous amount of work processing the more than 24 award applications. Using predefined eligibility and award criteria, the committee was tasked with providing a slate of recommended award winners across 5 award categories to the dean for his consideration. With such a pool of talented nominees, this was a difficult task, and I was truly impressed with their thoughtful review. And last but not least, on behalf of the New York Medical College School of Medicine, thank you to Dean Nadler. This initiative was your vision and a priority that you set for 2020. Now more than ever, we're appreciative of the opportunity to celebrate the achievements of our faculty. Ultimately, Dean Nadler selected 12 award winners and 6 faculty for honorable mention that we'll celebrate tonight. Now, without further ado, I give you Dr. Edward Halperin, Chancellor and CEO of New York Medical College, to introduce the first award category for excellence in diversity, equity, and inclusion.
Greetings to my fellow members of the faculty, and welcome to the program for the Dean of the School of Medicine's Faculty Awards. We have all lived through an extraordinary period of a sustained attack on the concept of expertise. That is, of course, what the professoriate is about, isn't it? It's about expertise. It is about people who choose to live in akademos, the place in Plato's garden where the lovers of knowledge dwell. And it is the responsibility of the professoriate to be engaged in the generation, conservation, and dissemination of knowledge. But we have just lived through a period of American history where there has been a sustained attack, as I said, on this concept of expertise. Why is it that some people are so incensed and attack the idea that other people just simply know more about something than you do, and you might be able to learn something from them? I can think of a few reasons why there are some elements in society who attack expertise. One, perhaps, is the abiding belief in fantastical thinking. People who get their primary source of scientific knowledge from their ministers, or who want to believe that we live in a world of fairies, leprechauns, and angels dancing on the head of a pin. I always think about that scary scene in the play Inherit the Wind about the Scopes trial regarding evolution in Tennessee, in which the song outside the window, "Give me that old time religion, give me that old time religion." It was good enough for mama, it was good enough for papa, it was good enough for grandpa, and it's good enough for me. So that's one source of the attack, I suppose, on expertise. Another is that people simply, to some extent, recoil from modernism. The world is too complicated a place, they say, and science and professors are dragging us into a world we just really can't handle, or we don't want to handle, or we don't want to think about. And so they recoil at the concept of expertise. I suppose the third reason is that experts sometimes discomfort the powerful. And when experts say things like poverty is a carcinogen, poverty promotes diabetes, hypertension, obesity, toxins in the atmosphere affect the health of people, smoking is bad for you, alcohol is bad for you. When we do things like that, we threaten how much richer rich people will get. And we interfere with their ideas of business and capitalism and profit. And so they strike back at us as pointy-headed intellectuals. Or the attack on Adlai Stevenson in 1952. He was an egghead. His speeches were too eloquent. And there are always some people who are simply convinced that they can figure things out by intuition, by, quote, common sense, by street smarts. You don't need an expert on foreign policy to discuss US foreign policy towards Russia, because I grew up in Alaska and Russia is nearby. So I know something about it. It's of course on its face absurd, but there seem to be some people in society who believe that, and for the past couple of years, many of them have been in positions of great power. And so they have recoiled from science, recoiled from expertise, and recoiled from knowledge, and we have all suffered as a society the consequences of that. So it is very worthwhile to have an event like the event today, and to remember the importance of what we do, the value of expertise, the strength of the professoriate, how much the faculty has to contribute to the university and to society, and to celebrate that, and to congratulate those members of the faculty who, in their pursuit of knowledge, have demonstrated considerable and laudable excellence. I thank Dean Nadler for including me in the program today, and congratulate all my colleagues who are about to become award winners at this Faculty Awards celebration. Have a good program.
Good evening, distinguished faculty, guests, family, and friends. First and foremost, thank you, Dr. Nadler, for holding this award ceremony to honor our faculty members who are doing exceptional work in so many areas here at NYMC. Your leadership and support is greatly appreciated. New York Medical College was founded in 1860, and 1 year after its founding, the United States was engaged in a civil war which went from 1861 to 1865. NYMC admitted its first woman during the Civil War and admitted the first African-American student just 4 years after the conclusion of that war, while the US was going through Reconstruction. As the world has changed, NYMC has remained consistent while embracing that change. NYMC has remained consistent as a bastion against bigotry as it continued to admit students who had been marginalized as we weathered through 2 world wars, the Great Depression, the Civil Rights Movement, wars in Korea, Vietnam, Iraq, and Afghanistan. This past year, 2020, the world became gripped by a global pandemic, and we saw the resurgence of the Civil Rights Movement here in the United States. As the world was changing, NYMC once again remained consistent while embracing that change. I was proud to see the high level of engagement of our students and faculty as we took the bold steps to institute an anti-racism and anti-bias in medicine curriculum. I thank Doctors Pam Ludmer, Ray Witt, Tracy Gardner, Jennifer Kessler, Jane Ponterio, Susan Racklin, Joanna Pesolano, Catherine Spunk-Nabel, Gary Stallings, Pam Lucchese, Pat Stanton, Amy Ansel, and others for their unwavering commitment to championing diversity and inclusion here at the NYMC School of Medicine. Thanks to Dr. Karen Murray and her admissions team for their holistic approach to admissions, which has been instrumental in making NYMC a leader in student diversity in our nation's medical schools. Thank you to Dr. Christina Peterson and her team from the Office of Academic Support for all the work they do to support our students and helping them to achieve success. Thanks to all those who have served as diversity and inclusion deans at NYMC, including Dr. Gladys Ayala, Monty Douglas, Camille Clare, Ms. Joan June. I look forward to building on their past successes. Thanks to the many student groups who have been consistent champions for diversity and inclusion here at NYMC, including the SNMA, LMSA, APAMSA, SAMHSA, AMWA, LGBTQ Advocacy in Medicine Club, and all the other wonderful student groups that work tirelessly to support diversity, equity, and inclusion here at NYMC. Indeed, the students here at NYMC are truly remarkable in their support of our efforts. As an example, the newly formed Culinary Medicine Interest Group has reached out to our office and are planning events in the realm of public health nutrition, as well as hunger awareness and advocacy. Special thanks to the NYMC Student Senate, who have worked very hard to hold our school accountable and have shown that they truly care about these issues. If you've ever had a conversation with me about diversity, equity, and inclusion, you know that I have a tendency to stress the importance of belonging. And you know, I always say people who feel they belong perform better at work, perform better in school, and have better health outcomes. I have been asked many times about why I have been so passionate about this area, and it all goes back to some of my early childhood experiences. My family immigrated from Haiti when I was 5 years old. I grew up right across the bridge in Rockland County. I still recall being a child in elementary school as HIV/AIDS was spreading across the country. There was widespread belief that one source of HIV/AIDS was Haitian people. I still remember one of my elementary school teachers separating me from the other children in the classroom because of concern that I might spread this infection, an infection that I did not have, to the other children. I know how I felt at that time, and I have vowed to do everything in my power to not let anyone else have that feeling of not belonging. And you better believe we are working very hard to make sure that our students, our faculty, and our patients feel included and that they belong. Despite all our great accomplishments, it is important that we are not to rest on our laurels, but remain steadfast in our mission of finding more ways to make sure that we have a truly inclusive environment where everyone can thrive and excel. I am truly humbled to receive this award for diversity, equity, and inclusion. I see this as a new beginning and a new day at NYMC. The world continues to change around us, and I know that we here at NYMC will remain consistent in fighting racism and bias while we embrace that change. Finally, I give thanks to my wife, Ainalum Etienne, and my children, Isaac and Grace, who support me throughout all of my endeavors. Thank you once again, and I wish you all a good evening.
Congratulations, Dr. Etienne. Up next is the Rising Star Award that recognizes 2 junior assistant professors from a basic and a clinical science department who excel in an area related to teaching, research, or institutional service. The inaugural Rising Star Dean's Award goes to Dr. Jonathan Fisher and Dr. Julia Kels.
I'd like to thank the Dean and the rest of the college for the honor of being nominated for this award. Science is hard. There are many hurdles you're dealing with the unpredictable forces of nature. And so it takes a team. One of the most important partnerships, the most fundamental ones, is between a principal investigator and the institution. It's because of my partnership with New York Medical College that we've been able to delve so deeply into science. And so that's why it's really an honor to be acknowledged by my partner in crime, New York Medical College. So thank you once again for this honor.
Hello, I'm Julia Keltz, and I'm honored and humbled to receive the Dean's Faculty Rising Star Award. Thank you, Dr. Nadler and the Dean's Faculty Award Committee for your consideration, and to the other nominees who have greatly contributed to our academic community. My grandfather always said, a patient should feel better walking out of your office than they did walking in, even if you gave them bad news. This has been a guiding principle for how I practice medicine and the empathy and compassion I teach to my residents and students. We care for patients at the most vulnerable times in their lives, and it is our responsibility to provide exceptional clinical and compassionate care to all. With this in mind, it's a privilege to contribute to the future of medicine here at New York Medical College and Westchester Medical Center in the Department of Obstetrics and Gynecology. I am passionate about elevating the care of women in our community and helping to create the generation of incredible physicians. I would like to thank Dr. Tejerati for his everlasting support of my career and unrelenting leadership in the department as a champion for women's health. Thank you, Dr. McCarrick, for your support and guidance, as I believe I am the only new residency program director in OBGYN in the country in the midst of a global pandemic. I would also like to thank Drs. Kara Grimes and Tana Pradhan, who have been my close friends and mentors over the last few years and encouraged the growth of women leaders in medicine and have laid the foundation for my developing career. Finally, I am most grateful for my residents who are eager to learn and become the future leaders in women's health, and to my patients who are the reason for everything I do. Thank you all.
Congratulations to Drs. Fisher and Keltz. The next award category recognizes faculty who excel in their work by consistently performing beyond expectations in teaching and in mentoring our undergraduate and postgraduate students, faculty, and colleagues. The Dean chose to broaden this category from its original 2 awards to 4, and also created a Distinguished Teaching and Mentoring Senior Faculty Award for this category. It is my great pleasure to announce the following individuals selected for excellence in teaching and excellence in mentoring. They are Dr. Brian Ratliff and Dr. Joseph Giamelli for excellence in teaching, and Dr. Raj Tiwari and Dr. Claude Andrew Powers for excellence in mentoring.
I would like to thank Dr. Nadler for this inaugural Excellence in Teaching Dean's Award. I would like to thank the awards committee for selecting me as well. I would also like to thank Dr. Leonard, the chairman of the physiology department, along with all the faculty of the physiology department for their continued support. In addition, there are numerous other faculty members and administrators here at New York Medical College that have given me unwavering support over the years. I want to thank each and every one of you. I would also like to thank all the students, both past and present here at New York Medical College, that have always been supportive of me. It is because of each and every one of you that I do this work. On a personal level, I would like to thank the good Lord who has blessed me with so much in this life. Without him, none of this would be possible. And finally, I would like to thank my beautiful and loving wife and children for all their support that they have shown me over the years, and the patience that they have consistently demonstrated as I have worked what at times has seemed like endless hours. Again, thank you. This is an absolute honor to win this award.
Thank you. I wanted to thank the selection committee and Dr. Nadler for presenting me with Excellence in Teaching Dean's Faculty Award. I know that New York Medical College gave me an outstanding education and more than prepared me for residency and my career as a pediatric cardiologist. It has been and continues to be an honor to educate and guide the students of New York Medical College toward phenomenal careers. I, of course, want to thank my mentors in education since I have been a student here, Dr. Leonard Newman, Dr. Michael Gowitz, for their support and guidance that led up to this award. I especially want to thank Dr. Paul Wolf, who unfortunately passed away from cancer in 2010. He was a true mentor and friend at every level of my career and personal life. I would like to receive this award also on his behalf. I definitely owe my passion for teaching and mentoring to him. And as always, I thank my wife, Anna, and children, Samantha and Michael, for their support in all my endeavors.
Thank you very much for bestowing this honor of excellence in mentoring. It is indeed a privilege to work at New York Medical College and be honored with this particular award. I would like to thank, first of all, the hundreds of mentees, both master's and PhD students that have clinical fellows, as well as junior faculty, as well as mid-level faculty that I have mentored, that have actually brought me to this position. And they are all now, a good number of them are in leadership positions and are doing very well in science. That actually gives me great pleasure. Second of all, I would like to thank Dr. Nadler for initiating this incentive and this award, and the committee for really recognizing me for this particular award. No award or the work of an award can actually be complete without colleagues that actually help you in shaping your research, as well as and mentoring is a collective effort. And in that respect, I would like to thank Dr. Gupta, who has been a wonderful colleague for almost about 24 years that we have spent together at New York Medical College. I would like to sort of in my address, I would like to really talk about what really constitutes a mentor. And this comes from a tradition that the Eastern tradition that I come from, which is the guru-disciple relationship, where basically the idea is for the guru in Sanskrit means one who dispels darkness. And it is one who imparts into the mentees or his disciples the ideas and the values of respect, dignity, and giving the space. The idea is that the disciple should, or the mentee should at least become like the mentor, if not more. Finally, I would again thank Dr. Nadler and the committee for this particular award, and all the different mentors and mentees that have actually shaped my experience. Thank you very much, and I'm really honored to accept this award.
Hello, everyone. First of all, I want to let everyone know how proud and pleased I am to have received the Mentoring Excellence Award as part of the Dean's Faculty Awards this year. It really means a lot to me. And I also especially want to thank Dr. Nadler for actually creating these awards. And also wish to thank Dr. Schwartzman, my department chair, who nominated me for this award. In addition, I also want to thank Dr. Spanknagel, as well as the other members of the other faculty members that were on the review committee for this award that actually selected me. I'm very happy about to receive this award. I also want to note that I'd like to also thank all of my faculty colleagues in the Department of Pharmacology, as well as in other departments that really have I've been interacting with in terms of my role, in terms of teaching and other roles in the New York Medical College. They have really played a major role in the success that I've achieved. And really, I am the course director for the medical pharmacology course. I've actually been in this role for over 20 years. If there's one thing that I have learned during that time is that I cannot do all the teaching on my own, and I depend tremendously upon the collaboration and the assistance of multiple faculty in order to achieve excellence in these activities. And one of the key things that I've always been trying to do in terms of my efforts in teaching is to bring innovation as well as continual change to meet the changing expectations of the students, as well as the changing student culture, as well as the changing requirements that are brought, we are told to comply with from the LCME in terms of medical education standards. This is something that takes a lot of thought as well as effort, not only on myself, but also in terms of getting all of the faculty involved, which is going to be has been very important, I think, for our success in the teaching that we've done, as well as moving ahead and looking into the future. And I can certainly say that one always has to be well prepared for what changes might arise, as we have learned so well in terms of the COVID-19 pandemic, and being able to actually continue our teaching mission for both myself as well as all of our faculty, which required a whole new level of technological innovation, which again is something that I've always tried to bring in to the medical pharmacology course in a wide variety of methods, I guess one could say, in terms of interactions with students, in terms of problem-solving exercises, to the daily active engagement in terms of having turning point audience response systems that's going to engage them even during large group sessions, et cetera. So again, all of that is something that I think is something that, again, has contributed to our success, and I would never have been able to achieve that without the assistance of my faculty that were also involved in this effort. So again, I'll just note that, again, I'm grateful for this award, and thanks again to the dean as well as all of the faculty that selected me for this award. Thank you very much.
Congratulations to the award winners. The next category, the Distinguished Award for Excellence in Teaching and Mentoring, as the only award category that was defined in retrospect. The inspiration for its creation and recognition goes to Dr. Kenneth Larea for his exemplary teaching, mentoring, and service over a decades-long distinguished career.
Thank you, Dr. Nadler and the selection committee. It is truly an honor to receive this recognition, especially for my peers, but it is an honor that must be shared with all my New York Medical College colleagues, who I've had the privilege to learn from and work with throughout the years. We all embrace the student body, and our graduates continue to make important contributions to the communities. We have a rich history of teaching faculty who have made NYMC the life, not just the livelihood. I arrived at the college in 1989 and was mentored by the many outstanding faculty who were essentially at the stage where I am today. They were the pillars of the college for many years. Our graduates will fondly remember each, and I apologize for only naming a few, but the mentoring by Drs. Argani, Dracontides, Grayson, Gorderi, Horowitz, Levine, Messina, Orlick, Passo, Wink, and Zakharow helped many of us to develop teaching skills that allow for transitions from yesterday's to today's programs. None of us teach in silos. It's the teamwork among our community that allows for successful teaching platforms. It is most enjoyable to collaborate with my many clinical colleagues and colleagues from both the Graduate School of Basic Medical Sciences and School of Health Science and Practice. It is unfortunate today we simply cannot roam the corridors of the Basic Sciences building and engage in discussions, but this will change. I thank all teaching faculty for opening the doors. Thanks to all program directors of the graduate school, and to Drs. Galipta, Lento, Olson, Kravitz, Peters, Powers, Thompson, Vilasek for being supportive, course-directive colleagues. Most importantly, these cohesive partnerships are what makes programs and courses successful. From the IT support of Michael, Alex, Nick, and Juan, to all the administrative staffs, in my case, Liza, Laura, Juliana, Cindy, Barbara, Valerie, Carolyn, to medical education, to my teaching colleagues and friends, Drs. Ettlinger, Fried, Kamarasuri, Peng, Newman, Sharman, and Zeman, who have been by my side since the beginning. Together, we continually implement changes to keep courses modern, to meet the needs of today's learners, with invaluable support from our colleagues at Westchester Medical Center and other departments, and from the insight and expertise from Jason and his colleagues in educational technology. We are proud to have wonderful students, and your feedback throughout the year has been invaluable to my own development as a teacher, and I thank you for that. Last, as I mentioned, NYMC has been a tradition of being the light for many of us. Over the years, we formed essentially a family bond. Dr. Frank Bellany, as Dean, created a collegial spirit among all of us in the graduate school and an atmosphere that welcomed creativity and collaboration among the many faculty, benefiting all students. His passion was contagious, and we often lost track of time. I thank my wife, Shelley, and daughter, Jackie, and stepdaughter, Dana, for putting up with the time I focused on the teaching and mentoring endeavors. Much thanks to Dr. Joe Etlinger, who fostered a student-centric environment throughout these years. But mostly, I thank him for recognizing the wonderful qualities in Connie and taking a chance on me, bringing both of us to the college as junior faculty. Together, we began as a team, and today we share in this honor. To all the graduates of the medical school and graduate schools, I think of you daily, and slowly I'm reaching out to reconnect. From California to Texas to Florida to Connecticut, you have my gratitude, for I learned from you when you were students and continue to learn from you when we speak. Who is the teacher and mentor? I would say you were and are. Thank you very much for this honor.
Congratulations, Dr. Larea. The next award category recognizes a basic and a clinical science faculty member who are emerging or established in their field as leaders in research. I'm pleased to announce the Dean's selection for Excellence in Research Award, Dr. Chandra Bakshi and Dr. Rafat Latifi.
Good evening. I'm extremely delighted to accept the Dean's Award for Excellence in Research today. I would especially like to thank Dean Nadler and Dean Spanknebel for this huge honor. As researchers, we know that it takes a village to publish a manuscript or receive grant funding. Any achievement in this field would not be possible without a strong team. And I would like to take a moment to acknowledge those that have strongly contributed to my success over the years. Firstly, I would like to thank my former chair, Dr. Ira Schwartz, and my current interim chair, Dr. Raj Tiwari, for their constant encouragement, excellent mentorship, and unconditional support that allowed me to establish a successful research program in my lab. I would also like to extend my thanks to my departmental colleagues, collaborators, and administrative staff, without whom this achievement would not have been possible. Finally, and perhaps most importantly, I would like to share this award with the highly motivated and hardworking postdoctoral fellows, PhD students, and master's students who have worked tirelessly in my lab to conduct quality research and publish impactful papers. I also offer my congratulations to all my fellow faculty members who are also being recognized for their distinguished achievement this evening. I feel really proud to be a part of this group. Thanks once again for this honor.
Dear Dr. Spanknebel, thank you so much for that very great news. And let me take this opportunity to thank Dr. Nadler and College of Medicine for this really important initiative to recognize faculties that do a lot of research, work with students, residents, and other faculties, both within their departments and as well as interdepartmentally. Clearly, I come to you via Zoom today on behalf of all of those who work in the Department of Surgery. Without the superb research team that we have been able to put together, without faculties, students and residents and research fellows, this will have never been possible. So I take a lot of pride on this award, but this one goes to everyone in the Department of Surgery. I am grateful again to everyone, and we stand ready to continue to do more research and enrich our research portfolio in defined areas of surgery, trauma, critical care, down wall reconstructions, oncology, and other disciplines within the surgery and beyond that. In addition, telemedicine and global health will have a prominent role in the research portfolio of the Department of Surgery. Thank you again, and have a great day.
Congratulations, Drs. Bakshi and Latifi. I'm excited to introduce the next and final award category, which is the Dean's Highest Distinction. This inaugural year, 2 faculty were selected for the Dean's Outstanding Achievement Award. This award recognizes a senior faculty member whose efforts have sustained significant achievement in all areas of teaching, research, and service. We've invited each awardee to provide a brief overview of their research and its impact that combined encompass more than a half-century of scholarly work at New York Medical College. Congratulations to Dr. Mitchell Cairo and Dr. Gary Wormser.
Good evening, and I would like to first say that I'm very humbled and honored for being awarded the Dean's Outstanding Achievement Award. I particularly want to thank Dr. Newman, who nominated me from the Department of Pediatrics, and thank Dr. Spanknebel and the review committee, and of course, thank Dean Nadler for this honor. Oops, okay. So it's kind of hard to cover in 15 minutes, my career achievements. So I've kind of summarized this on the outline. And these are the topics I'm going to try to cover quickly to show you the breadth and the depth of what we've accomplished. And this actually is my 10-year anniversary here at New York Medical College. And I want to end with a couple of words about mentorship and acknowledgements. So just very briefly, we have spent a lot of time and effort in trying to understand the processes that go on in immunology and stem cell regeneration, starting from the embryo all the way out to peripheral blood. And I think our first major achievement was in the area of cord blood. So approximately 20 years ago, received a grant from the National Heart and Lung and Blood Institute to develop the first federal cord blood bank. I did this in collaboration with Dr. Kurtzberg and Dr. Frazier, where we established 3 cord blood banks. And this is where we established the methodology and the standard operating procedures for cord blood collection, cord blood processing and cryopreservation, and ultimately cord blood thawing and administration. And through this process, we were able to establish the incredible enrichment that occurs in cord blood and hematopoietic progenitor cells that's shown here, both in terms of colony formation and certain CD34 subsets. And then we importantly identified for the first time the log relationship between total nucleated cells and the log of CD34 and the relationship of CD34 and progenitor cells. And this now allowed us to launch into cord blood transplantation. And our first landmark paper that we published showed that we can use unrelated cord blood to reconstitute children with a variety of malignant, non-malignant disease. This was the first publication of its time. And this I did in long-term collaboration with one of our senior scientists who's still with me after 37 years, Carmela Vandevam. This led to the whole process of cord blood banking. And as you can see here, there are 3 options with cord blood collection. One is the usual discard. One is for public banking, which I had shown you earlier. And then that has led now to private banking or directed banking that can be used either for a sibling or for a directed donor in the family for the patient themselves. And that's then led to my participation in the publication of 3 American Academy of Pediatrics statements on the role of cord blood banking. So moving on to the second topic of achievement is tumor lysis syndrome. Briefly, this is an oncological emergency that's related to the breakdown of tumor cells that lead to the release of a number of metabolites leading to acute kidney injury and cardiac failure. And that's related to a number of risk factors that we've established. In this area, then, we went on and established the diagnosis and grading of tumor lysis syndrome, as shown here, which is still in effect today after some 16 years. And that led then to the pivotal trials where we were able to demonstrate that a new drug called rasburicase was able to break down uric acid, converting into allantoin, which is 20 times more soluble than uric acid. And we did the pivotal trial showing the comparison of rasburicase versus the standard treatment of allopurinol, showing that could rapidly and more consistently lower uric acid. And then we later showed this in the Children's Oncology Group trial, showing that we can prevent the progression of tumor lysis syndrome. That segues into the work we've done in B-cell lymphoma. This cartoon showing you the various forms of B-cell lymphoma. The most common that occurs in children is Burkitt's lymphoma and diffuse large B-cell lymphoma. And we've done a series of studies over time, which shows that we've been able to double the outcome in patients with Burkitt's lymphoma by using a protocol that we developed that used short intensive chemotherapy. And this is shown here where we performed the first international collaboration in collaboration with Catherine Pott, shown here, who was head of the French Pediatric Oncology Group, where we tested this new approach of short and intensive therapy. And this is shown in several publications that we had as the first international pediatric oncology protocol performed in 3 international pediatric oncology groups, where we were able to now improve the outcome in low risk disease to 99%, intermediate risk to 90%, and now in high risk disease, 88% survival. In collaboration with Sherry Perkins from the University of Utah, we were able to demonstrate in all forms of mature B-cell lymphoma in children that they overexpressed CD20, and an antibody directed against CD20 is a drug called rituximab. In collaboration with Stan Goldman, a long-term colleague and former faculty member of mine, we're able to now combine rituximab with the same chemotherapy background I showed earlier, and now raise the survival rate in the intermediate risk group to 95% and the high risk group to 90%. And more recently here at New York Medical College and Westchester Medical Center, in collaboration with Jess Hochberg, an assistant professor in our program, and Leanna Klagemont, a research assistant professor, we now were able to reduce the doses of some of the chemotherapy regimens and develop a new protocol called Reboot. And this is the data we just presented last month, the American Society of Hematology, showing now 100% survival by the use of chemoimmunotherapy. The next achievement I want to quickly cover is the work we've done in sickle cell disease. This is an autosomal recessive disorder. It's an orphan disease. It tends to be found in African American and Latino patients. And its complications include chronic organ failure, such that patients usually die in their mid-40s. And we've been unable to find a cure for this other than allogeneic transplant. Unfortunately, only about 15% of children have a brother or sister that's a tissue match that also don't have sickle cell disease. So through a lot of work over the last 20 years, we were unable to show that we can achieve a high cure rate with unrelated donors, both adult donors or cord blood donors. So we began to move into a haploidentical donor. And here now at Westchester Medical Center, New York Medical College, we developed a processing system where we can enrich the cells for hematopoietic progenitor cells and add a small amount of immune cells and use a mother or father as the donor with a myeloimmunosuppressive conditioning regimen. And what we've been able to show, besides the high cure rate, is that we're able to show stable neurological and significantly improved health quality of life. In collaboration with Dr. Brannicky, assistant professor in our division, and in collaboration with Dr. Flower in our division, another assistant professor, you can see that we can achieve a 90% one-year event-free survival using a parent as a donor using this methodology. And this now provides the same type of cure rate that we see when we use a brother-sister tissue match donor. Moving on to the work that we've done in solid tumors very quickly, in collaboration with Dr. Rosenblum, shown here, assistant professor in our division and the leader of our sarcoma program. We know that the outcome in patients with metastatic disease at diagnosis do not do well, and new therapy is required. And Dr. Lowe in our division, who's an assistant professor in pediatrics and cell biology. We have been working on trying to find a downstream target, which in this case is PP1R1A, which is a downstream target of EWS fly, which is the common translocation found in Ewing sarcoma. And using a variety of approaches, both a knockdown as well as using inhibitor, we're able to show a significant improvement in the outcome of animals that have been xenografted with Ewing sarcoma. And this is allow us to now provide a new pathway of trying to cure which has been an uncurable disease. And similar work with Dr. Lau, as well as Dr. Garden Schwartz, an assistant professor. We've also looked at an immunotherapy approach where we have identified a target called MCAM, or otherwise known as melanoma cell adhesion molecule, which is highly expressed in Ewing sarcoma. And now we've developed a chimeric antigen receptor that we've been able to transduce into NK cells in combination with a new IL-15 receptor agonist. We're now making strides in hoping to bring this to the clinic to treat refractory metastatic Ewing sarcoma. Now, moving on to the next topic quickly, and work that we've done is try to find a way of identifying both hematopoietic progenitor cells and immune suppressor cells in both cord blood and placenta. This is work that I've done with Dr. Liao. This work we've done is concentrated in a genetic disease called recessive dystrophic epidermolysis bullosa, which is missing the collagen 7A gene, which is shown here in the RDEB patients versus the normal wild type. And very quickly, the work that we've done using human placental-derived stem cells in collaboration with a company called Cellularity. We've been able to make progress using a collagen 7A knockout, where we've been able to prevent the complications that normally occur and show the demonstration now of new collagen 7A formation. And then in a series of studies that we've done together, which has made the cover of Cell Transplantation Stem Cells and Stem Cell Translational Research, we're able to show that cord blood derived unrestricted stem cells, another product that we get from cord blood, we can detect these cells migrate to the wounds of animals with RDEB. We can suppress the hemorrhagic blistering that occurs, and now we can show that they engraft in the GI and skin tract in these knockout mice. And more recently, in collaboration with our neonatal group, particularly Dr. Gama and Dr. Vinakunda, we've been able to then apply these USSCs to show that we can now suppress ventricular enlargement, show that they can go to the brain in babies, in this case in an animal model, who developed intraventricular hemorrhage and hydrocephalus. Now, moving on to the last couple of achievements. This is work that we've done in trying to expand natural killer cells. Initial work with Dr. Aiello, or research assistant professor. In our division, we used a bunch of cytokines to expand cells. And more recently, in collaboration with Dr. Chu, an assistant professor in our division, we have developed a genetically modified feeder cell, which can expand the NK cells by 30 to 40,000 fold. And Dr. Liao and I have developed a method of in vitro transcription of a chimeric antigen receptor DNA into an mRNA, and using electroporation, we can now transfect these cells with the CAR mRNA. And showed here on the left, we show significant killing compared to the mock transfection. And in a xenograft model, we can show significant enhancement, reduced tumor burden with these CAR mRNAs. And now shown more recently with the use of a histone deacetylase inhibitor, we now get significant survival in animals that are xenografted with Burkitt's lymphoma using a CD20 CAR mRNA. And the last work that we've done that was just published this year in the Journal of Immunotherapy for Cancer, we've taken an IL-15 molecule that we've now superimposed the FAB binding site to CD20. So we now have an immunocytokine where we have an IL-15 and a CD20, and we can show significant killing in a CD20 Burkitt lymphoma xenografted model, which works a lot better than using the native rituximab and the IL-15 binding molecule. And then lastly, we've done work with developing cytotoxic T lymphocytes against refractory viral infections that occur particularly in the allogeneic stem cell population in patients with primary immunodeficiency. This is where we will take a donor and we'll test them whether they respond to the peptide of that particular virus. And then we can go ahead and manufacture, if they respond, and enrich cells from a donor that have a memory specifically to that particular virus. We're doing this under a grant we have under an R01, where we're testing CMV, adeno, EBV, and BKV CTLs in patients. This is showing the CTLs that we're able to generate with adenovirus, BK, and CMV. And in the initial studies we've done so far, we have a complete response rate of 83% and overall response rate of 100%. And this is segued into new work we're doing with SARS-CoV-2, where we take the same processes where the spike protein, the M protein, the N protein, we use specific peptivators to 3 of those proteins. And in work that Dr. Milner, assistant professor, and Dr. Rigaud in our CTEL lab, among others, we've now been able to generate SARS-CoV-2 CTLs, both CD8 and CD4. And we now submitted a protocol to the FDA, which we're waiting to hear on in the next couple of weeks, to do a first-in-man study using family members who have already had SARS-CoV-2 and be able to give them SARS-CoV CTLs that we've generated through this process. I'd like to end with the mentorship part of my career. This just shows you in 2 slides some 45 fellows that I've trained and where they are right now, basically all over the planet, so to speak. And it's been my pleasure to participate in, and some also 50 residents that I've trained. And I think I've acknowledged by picture at least most of everybody on the slide. I do want to mention specifically Aaron Morris, my 40-year research administrator, Lauren Harrison, some 23 years as our lead clinical research nurse, and Miguel Muniz, who's been with me 20 years as my administrative assistant. And thank the leadership at the institution, particularly Dr. Newman and Dr. Gowitz, who have been very supportive, that's allowed me to be eligible to receive this achievement award. And it is, of course, it takes a village to accomplish this. This is a recent picture of most, but not all of the members of our program. And last but not least, thank my family. This upcoming Valentine's Day will be my 50th anniversary of my first date with my wife, Ellen, who's been a big supporter and my biggest fan. And my 2 sons, Dustin and Evan Cairo, who have also allowed me to take time to do this. And my recent daughter-in-law, Alana, who's a clinical psychologist at the Manhattan VA. and thank all of our grant supporters over the years that make this possible. Thank you.
So I would very much like to say how honored I am for getting the Outstanding Faculty Award for 2021. And I want to very much thank Dr. Nadler and Dr. Sugar for nominating me. I'd also like to thank the award committee headed up by Dr. Spanknebel and all my coworkers and collaborators who were so invaluable to the success of our research efforts over these many years. A partial list of people I've worked with and who've been critical to the success of our research endeavors as shown on this slide. It's too numerous to count, and I don't have time to list everybody, but I would like to mention specifically Dr. Nadelman and Dr. Ira Schwartz. In 1977, I finished my infectious disease fellowship at Mount Sinai Hospital and stayed on as an attending physician at Mount Sinai and at the Bronx VA, an affiliated hospital. In June of 1980, a patient was admitted to the Bronx VA hospital who had been sick, a young man who had been sick for about 2 to 3 months with cough, fevers, and weight loss. His chest x-ray is shown here that shows bilateral interstitial infiltrates, very atypical for community-acquired pneumonia. And so a lung biopsy was done that showed that the patient had pneumocystis carinii pneumonia. But pneumocystis carinii pneumonia we had never seen as infectious disease doctors in patients without known underlying immunocompromising diseases such as an organ transplant. So from the very first case, that something new was going on. And I and my other colleagues who were seeing similar cases around New York City recognized this immediately. And we got together under the leadership of Dr. Henry Mazur to work up the immune system immunologically of these patients that were being diagnosed with unexplained pneumocystis carinii pneumonia. We had 11 patients who met this criteria for inclusion in the study. It turns out they were all men and they had pneumocystis carinii pneumonia. Of interest, they were all young men. They were of diverse ethnic background, and about half were intravenous drug users, and about half were men who had sex with men. A very distinctive epidemiologic pattern, and very much the pattern that was being seen in the early days of the AIDS epidemic. In December 10th, 1981, there were 3 publications in the New England Journal appearing back to back, in which was described a new acquired cellular immunodeficiency disorder, such as what our patients had. And our patients are included among the 3 publications. It turns out that these were the first peer-reviewed publications in the medical literature describing a new acquired cellular immunodeficiency disorder associated with opportunistic infections. A little closer view of one of the publications is shown here. And this is me. And this is of all the publications I've had over the years, I'm most proud to be co-author of this publication. And it was all based on an immediate evaluation of the first cases of unexplained pneumocystis carinii pneumonia that we had, in which we found that they all had a very similar cellular immunodeficiency state disorder. So other AIDS research that I participated in, some of the examples are shown on this slide and the next slide. When I joined New York Medical College in the spring of 1981, we were then affiliated with Lincoln Hospital, and I was made aware that there was a woman who had been admitted to Lincoln Hospital who also had pneumocystis carinii pneumonia, but again, without any explanation or underlying disease that would have explained it. So I immediately called up Dr. Henry Mazur and said, why don't we enroll patients that are women who have also unexplained pneumocystis carinii pneumonia? He agreed, and we enrolled that patient and 4 other patients around New York City. and showed that they had a similar acquired cellular immunodeficiency disorder, which has subsequently been called AIDS, of course. And this publication was in the Annals of Internal Medicine in 1982. And it turned out the women were either intravenous drug users or sexual partners of an intravenous drug user. Because the study was my idea, I moved up somewhat in the author list. Another study that we began at Westchester Medical Center in 1982 was evaluating healthcare workers who had needle stick accidents with AIDS patients. And this is, of course, quite concerning to the healthcare worker because of the potential risk of blood transmission of this virus that clearly could be transmitted by blood and sexually. And so I developed a collaboration with Dr. Gallo at the NIH, who was working on developing a diagnostic test to detect HIV infection. And we sent him the blood specimens that we had collected serially from our prospectively enrolled subjects. And similarly, Dr. Marty Hirsch at Mass General was doing the exact same study and also collaborating with Dr. Gallo. When Dr. Gallo got the results finally, he suggested that Dr. Hirsch and I collaborate and put our results together in one publication, which we did, that was published in the New England Journal in 1985. Even though it was clear that, based on the epidemiology, that AIDS wasn't transmitted through casual contact, many people still had the notion that it could be. And they wouldn't shake people's hands, and they were very nervous about athletes participating in sports because of the potential, they thought, of sweat being infectious. So at New York Medical College, we did a specific study to address the question of whether HIV was in sweat, looking at both cultures of sweat and PCR for detection of DNA and RNA in sweat samples. And we found no evidence whatsoever for HIV being in sweat. However, because Lyme disease and tick-borne diseases are very important in our general geographic area, we also started working on research projects related to Lyme disease in the mid to late 1980s. From 1987 to 1996, we enrolled approximately 500 subjects into a randomized, prospective, placebo-controlled, double-blind study evaluating the use of a single dose of doxycycline given within 72 hours of a deer tick bite to prevent Lyme disease. The basis for this study is that incubating infections, where there are much smaller numbers of organisms compared to overt infections, are more likely to be treatable with very short courses of antibiotics. And this study was published in 2001 in the New England Journal with an accompanying editorial. In our 2001 study that I just mentioned, there was an 87% efficacy rate of the single dose of doxycycline. Indeed, subsequent to that study, single dose doxycycline is now commonly used to treat patients after a high-risk tick bite in the United States. Motivated in part because of our study, a similar study was conducted in Europe, also of a single dose doxycycline within 72 hours of a tick bite. And similarly, it was a single dose doxy was found to be 67% effective. In a French study, based on our study in part, looked at the same issue after high-risk sexual events in terms of potentially preventing syphilis. And similarly, they found efficacy of 72% in preventing syphilis after a sexual event when the patients were given a single dose of doxycycline within 72 hours. In an Israeli study of people entering into caves for prevention of tick-borne relapsing fever, a single dose of doxycycline was found in a 2020 publication to be 100% effective. Other noteworthy Lyme disease research activities that we've been doing over the years was focused on use of culture. The reason we did that is culture is the gold standard for diagnosing infections and hadn't been used very much for diagnosing Lyme disease, leading to potential ambiguity as to the accuracy of the diagnosis. From 1991 to 2000, we enrolled 283 culture-confirmed subjects with Lyme disease into a long-term 20-year prospective study. As part of the study, we obtained isolates of Borrelia burgdorferi, and this enabled us to discover strain diversity and its consequences clinically. A couple of publications that I'd like to point out. One, whose first author is a medical student named Erica Weitzner, who was invaluable in helping us aggregate the data for our long-term outcome study. And the second one is on the issue of whether when a patient gets a second episode of early Lyme disease, is it a relapse of the original infection, or is it just due to another tick bite and reinfection with another strain of Borrelia burgdorferi? And this article, this study, which was published in New England Journal of Medicine, clearly showed, based on the strain differences, that recurrences of Lyme disease were generally reinfections, and in no way were they relapses from a prior infection. That last study was acknowledged as in the award ceremony of the New York Medical College Library, and Dr. Ira Schwartz, who did the strain diversity genetic testing, Dr. Nadelman, who was the first author of the study, and myself are shown in this picture. Thank you very much.
Thank you, Dr. Cairo and Dr. Wormser, for your truly inspiring work. This concludes the inaugural Dean's Faculty Awards virtual ceremony. Thank you for joining myself and Dean Nadler for this celebration, and congratulations to all the award winners. We hope to see you in person in the near future and wish you all a safe and healthy year ahead. Good night.